Hey, today we're going to be computing the individual counts of nucleotides within a DNA string. As a reminder, these four nucleobase molecules are adenine, A, cytosine, C, guanine, G, and thymine, T. First, let's create a sample DNA string composed of symbols representing the four nucleotides. Next, let's define the nucleotide count function and set it to have one parameter, the DNA string. As we iterate over the DNA string, we'll need some form of data structure to store the nucleotide counts. For this, we'll import default dicts from Python's built-in collections library. Then, we'll create a default dict with an integer data type. Using a default dict instead of a regular dictionary means that a default value, in this case the integer 0, will automatically be generated whenever a key is accessed. This will save us from having to initialize the counts to 0 for each nucleotide. Now that we have a variable to store the counts, we can iterate over each nucleotide of the string using a for loop. We will use the nucleobase symbols as the keys for the dictionary so that we can simply increment the respective counts. To wrap up the function, we'll return the counts dictionary. We can now go back and call this function. We'll also pass in our previously defined DNA string as the argument. Finally, we'll save the return counts in the variable and output them to the console. Although this problem only required a few lines of code, applying the power and scale of computing towards more challenging bioinformatics problems will prove to be very exciting. I hope you stay tuned for more, and thank you for watching.